see if that works better. That doesn't help?
It's lagging a second. Let's hear it. Let's wait.
I think I may have had it plugged into the line in rather than the mic. All right, so got it. All right. How late are we? Oh, shoot, we're late.
Oh, okay.
All right. I get this. Let me do one more thing here. I'm behind. Okay, to begin. So last time we had gotten as far as DNA synthesis, that is replication. Talked about how we could take a strand of DNA and replicate it. And the ideas there, the main concepts were the ideas of semi-conservative replication, the idea that the DNA polymerase could only read in one direction, 3' to 5'. That meant that gave rise to a leading strand where the DNA polymerase is reading right along the strand from one end to the other, theoretically, and making a complementary DNA strand. So that's the replication. The problem was that the DNA synthesis, the DNA polymerase, could only read in one direction, but that the two strands of DNA are oriented in opposite directions. That's the concept of the anti-parallel organization. That gave rise to a problem for the DNA polymerase. What it meant is that the DNA polymerase had to wait for the helicase to separate the strand for some distance, maybe a few hundred base pairs, before on that other strand, which was headed from the 5' to 3' end, wait for that 3' end to open up, apply that DNA polymerase at that region, and read back again. Some of you may have heard of the term Okazaki fragments. That's where this term originated. It originated in the laboratory of a guy named Okazaki in Japan, and he was interested in DNA replication. He was doing these experiments, and he was stopping the replication at certain sites. And he would find this odd arrangement where he would get great long strands of newly synthesized DNA and then shorter pieces. It was the shorter pieces called Okazaki fragments which led people to recognize that you could have a leading strand which you read continuously, but a lagging strand was then synthesized in fits and starts. And those are the origins of the so-called Okazaki fragments. What we're going to spend most of our time on today, though, is we're going to spend our time on the idea of a related nucleic acid. DNA is clearly a nucleic acid, but there's a related nucleic acid called RNA, ribonucleic acid. As the name implies, the ribonucleic acid has its basis in the 5-carbon sugar ribose, and we'll again review this idea of the 5-carbon sugar structure. We'll see that, as yesterday with the DNA, that there are 4 nitrogenous bases. There are 2 purines and 2 pyrimidines. The 2 purines are adenine and guanine, and the 2 pyrimidines now are cytosine and uracil. Now, this has a different name from thymine. Remember, yesterday was the thymine was the other pyrimidine, and thymine is the pyrimidine, one of the 2 pyrimidines in DNA. But now we see uracil, and what I want to emphasize here, and I'll show you a picture, is that thymine and uracil are remarkably similar. That in fact, thymine is often named biochemically as 5-methyl-uracil. So all we do is take that uracil, add a methyl group to it at the number 5 position, and we've converted uracil into thymine. Just to point out that these are very, very similar molecules, and yet that difference of that methyl group has a big change and a big effect on DNA versus RNA. Now, the other commonality here that we'll see, remember yesterday we spent a lot of time emphasizing how the deoxyriboses were linked one to the next into a great long polymer by phosphodiester bonds. Well, we can do the same thing. It'll take a different enzyme. It's going to take an RNA polymerase rather than a DNA polymerase. But the trick is going to be pretty much the same. We're going to form phosphodiester bonds between adjacent ribose molecules. So we can make this great long polymer of ribose molecules, and each of those ribose molecules, as we saw yesterday for the DNA, at the number 1 position, will have one of these 4 bases attached. So the story runs in parallel with only subtle differences. But the subtle differences become important. That lack of a hydrogen on the deoxyribose, or if you will, the addition of the hydroxyl to the number 2 carbon on the ribose, changes the ability, it changes the steric interaction of the sugars in such a way that, in contrast to DNA, which can be thrown into that regular repeating pattern of the double spiral helix, we find that for RNA, we don't get the molecule thrown into this regular pattern of the double helix. Instead, we say that it is a linear molecule. It's a single-stranded molecule. It doesn't base pair as DNA does. There are probably a couple of reasons for that. One of them is the lack of the hydroxyl in deoxyribose. And we put this great big hydroxyl group in ribose, and that probably in some way sterically prohibits the molecule from undergoing that regular pattern of the double helix. There's also probably a contribution to the methyl group on the thymine, which probably also contributes to that regular repeating pattern of the double helix in DNA, which is lacking in RNA. Now, having said that, this is an important concept that still arises, in that in RNA, we still can get some base pairing. It's not that regular pattern of base pairing that we see in DNA, but in fact, we can get some base pairing such that this RNA molecule can fold back on itself. And as the RNA molecule folds back on itself, it begins to generate a certain degree of 3-dimensional structure. Whenever you have 3-dimensional structure in a molecule and you have this difference in charge, which the bases can provide, you begin to see that you get rudimentary form of enzymatic activity in these RNAs. In fact, there are a number of RNAs that in fact act as enzymes. And one of the big ones we'll see later on, probably tomorrow or maybe on Monday, is the RNA in ribose. I'm sorry, the RNA in ribosomes. The RNA in ribosomes, will, particularly in large ribosomal subunit, will actually act as an enzyme that forms the peptide bonds. But before we get to that, we really need to think about how we're going to synthesize RNA. And of course, the RNA is going to be synthesized from a DNA template. So the DNA provides a template from which we'll synthesize a molecule of RNA. But if you think about that for a moment, you'll recognize that when we talk about a DNA molecule, we're talking about a chromosome. And that since we only have 46 chromosomes, 23 pairs, that means all 23,000 genes that we have in our genome have to be on those 23 chromosomes. What that means then is we have these long stretches of DNA which will run base to base to base to base, and within all of those bases there will be regions that will be the genes. There may be several hundred genes in a chromosome. So one of the issues we have to address is how do we go from this continuous string of bases in DNA and then find within that subregions from which we can transcribe the DNA into the RNA. The regions we call a gene will be transcribed into RNA. So how do we manage to take this great long string of bases and then find the right sites to say, start here, make the RNA? And we'll talk about that in some detail. And to do that, we need some definitions. There's a definition, the first one I'm going to tell you about is a promoter. A promoter is a set of bases in the DNA to which transcription factors bind. Usually this is a consensus sequence of bases. The most common of all is the Tata, known as the Tata box. Usually it's Tata followed by a couple of other purines. But there are a number of other promoters. That's simply the most common. What they are is a sequence of bases, a consensus sequence of bases to which transcription factors bind. Well, that raises the question, what's a transcription factor? Well, a transcription factor is a molecule, it's a DNA binding molecule that can recruit a whole assembly of proteins that will in turn recruit an RNA polymerase. So the promoter's in the DNA. It then binds to a transcription factor. The transcription factor on the DNA recruits a bunch of other proteins, an assembly of proteins that in turn recruits an RNA polymerase. The RNA polymerase can then read along the DNA strand, the template strand, again 3' to 5', reading along that DNA strand, producing a complementary molecule of RNA. Yesterday's emphasis was on replication, DNA to DNA. Today is on transcription, which will be DNA to RNA. So we'll read the DNA template and we'll make an RNA molecule. Now I'll finish with the idea that there are 3 major kinds of RNA. Actually, there's a 4th that we'll talk about briefly. The 3 major types that we're going to be interested in here, though, are messenger RNA, which encodes for the sequence of amino acids in the resulting protein, a transfer RNA, which acts as a kind of shuttlecraft, which is going to be carrying specific amino acids into the 3rd structure, the ribosome. The ribosome is made of ribosomal RNA, and the ribosome is where the peptide bonds are going to be made. And we'll talk about this mostly tomorrow when we bring these 3 characters together, messenger RNA, transfer RNA, and ribosomal RNA, into the process known as translation. Today, though, we're dealing with that part of the central dogma called transcription. Let me start by showing a couple of slides. Excuse me. Start by showing a couple of slides reminding you where we were yesterday and bringing home this idea of the leading and lagging strands. This is much of what we discussed yesterday. And the upper panel that shows this 5-carbon sugar, there's only a hydrogen here, 2 hydrogens, and the number 2 carbon, so this is deoxyribose. The number 1 carbon here is linked. What they're showing at this juncture is an adenine. Then there's the base pairing here between the adenine and the thymine. This is one of those pyrimidines, thymine, and then it's in turn connected by a covalent bond to the number 1 carbon on the deoxyribose. Again, we can then form, we can then form these phosphodiester bonds between adjacent deoxyribose molecules and make the backbone of the DNA molecule. And what we show here is the backbone of the DNA molecule. We're showing the adenines bind to thymines, cytosine to guanine. This is the base pairing, showing the hydrogen bonds, holding these 2 bases together. Then we finished with the idea of how we make copies of DNA molecules. And we finished with the idea that we would use the 2 old strands as template strands, and from that we would synthesize a new strand that's complementary to the old strand. But there were a couple of flies in the ointment here. The first was that on the leading strand, where we're reading 3' to 5', we can walk continuously along the strand and synthesize the new strand from the old template strand. But over here on the lagging strand, we had another problem because of the anti-parallel organization of the DNA molecule. If this is 3' to 5', then this one is 5' to 3'. And that's a restriction because the DNA polymerase can only read in one direction. So where on this strand it can read continuously on this strand, it has to wait for the helicase to open up the two strands, separate the two strands, and then it has to wait for maybe a few hundred base pairs to open, and then the DNA polymerase will arrive at this strand and read again in this direction. And once it gets to this point, it has to be annealed to make the two strands continuous with some other enzymes. But just to hold on to this idea that we have a leading strand and a lagging strand, and the Okazaki fragments are just an experimental artifact of when Okazaki was trying to sort out how DNA was replicated, and he found these little chunks called Okazaki fragments, and those were the chunks that were being read on the lagging strand. So any questions on that? Okay. If not, let's now begin thinking about RNA. I think it's worth thinking and concentrating on the idea that RNA and DNA are both nucleic acids. They're highly related molecules. They're quite similar to one another. But differences in their structure make them do or allow them to do different things. So we're going to start out here with ribonucleic acid. This, of course, is what we abbreviate as RNA. The components of ribonucleic acid are first the 5-carbon sugar, ribose, and then 4 bases, 2 of which are purines, adenine, guanine. These are the purines. And you'll recognize these as the same bases we had yesterday in DNA. The other 2 bases that, again, will be pyrimidines will be cytosine, again, the same as in DNA, and uracil. And these are the pyrimidines. Although uracil has a different name than thymine, it's really, really similar. Thymine is said to be 5-methyl uracil. Let me take a moment and show you that. Of course, don't memorize the structure here, but I like to show it because here's thymine. We'll count them here. 1, 2, 3, 4, 5. 1, 2, 3, 4. Right. 1, 2, 3, 4, 5. Here's the number 5 carbon. Number 5 carbon with thymine has this big, bulky methyl group, carbon, 3 hydrogens. Whereas in uracil, again, it's the same structure. The only thing that's different is, again, counting 1, 2, 3, 4, 5. This number 5 carbon now, rather than having a methyl group on it, has a simple hydrogen. That's the only distinction between thymine and uracil. So very, very similar molecules, both being pyrimidines and not so different from one another. But it turns out that these subtle differences between DNA and RNA are important. So let's now begin thinking about how we're going to organize this. And we'll go to the right-hand panel. And here we'll label this as the structure of RNA. And we'll follow the same pattern we had yesterday, where we have this 5-carbon sugar. 1, 2, 3, 4. And up here in the number on the flagpole is number 5 carbon sugar. As with yesterday, that number 5 carbon has on it an oxygen, a phosphorus, an oxygen, a phosphorus, an oxygen, and a phosphorus. And I won't go through all of it, but there's a double bonded oxygen and an O negative on each of the phosphoruses. But that's a triphosphate. And then out here, at the number 1 carbon, we can put a base. And I'll point out that one of the major distinctions between ribose and deoxyribose, in fact, the only distinction, is here at the number 2 carbon on ribose, we have a hydrogen and a hydroxyl. So put this big bulky group in at this point. But at the number 3 carbon, we still have the same old arrangement, in that we have a hydroxyl, we have a hydrogen. Again, I think you can begin to see where we're headed with this. We number the carbons, 1, 2, 3, 4, this is 5. It's going to make this the 5' end of the molecule, just as we saw yesterday with DNA. And then the next structure here is going to be another 5 carbon sugar. Again, it's going to have a base here. It's going to have a hydroxyl down here, a hydrogen, an OH, and an H. And here in the number 5 carbon, there's going to be a hydrogen, a hydrogen. And then out here, again, is going to be our phosphate group. So this is going to be O, P, O, P, O, P. I won't put all the other oxygens in. Now let me point out, while we're here, that if this structure, this ribose molecule, had out here at this number 1 carbon an adenine, this structure by itself would be adenine triphosphate. That's ATP. If this were a G, this would be GTP. Those are molecules that are very active in all of biology. And each of the bases can have this triphosphate. Adenine, guanine, cytosine, and uracil can have all of these triphosphates. Just as we saw yesterday, what we're going to do is we're going to use the energy in this diphosphate. Use the energy in that diphosphate. We're going to cleave off that diphosphate. And we're now going to form a new bond. Erase that. And we'll form a new bond, much like we did yesterday, where now we have the carbon, oxygen, phosphorus, carbon bond. So there's the carbon, there's an oxygen, there's a phosphorus, there's an oxygen. And you'll recognize this again as the phospho diester bond. So just as yesterday, we linked the deoxyriboses together in this great long string through those covalent linkages called the phosphodiester bonds, we can do the same thing here with RNA. Now we'll see here in a little bit it takes a different enzyme. It takes an RNA polymerase, where yesterday it was DNA polymerase. But nonetheless, we can make this great long string of ribose molecules. And again, form this phosphodiester bond. And I won't go through this over and over again. We saw it yesterday. We're seeing it again today. And what we're doing is we're generating this great long polymer of riboses, just as we did yesterday. It will have a 5' end, and down here it will have a 3' end. So again, we can talk about the orientation of these molecules, 5' ends and 3' ends. Now, yesterday, one of the big pieces we talked about was the fact that when we were using deoxyribose, because we lacked this hydroxyl down here, instead had only a hydrogen, and because we were using thymine instead of uracil, we could get this regular pattern of pairing, base pairing, in the middle of the molecule, that the 2 strands of the DNA would be held together by the base pairing, and that was a remarkably stable molecule. Because of this hydroxyl group, and probably because of the variations in the uracil versus thymine out here on the base, we can't form this molecule into that regular repeating pattern of the double helix. Instead, we say that RNA is a single-stranded molecule. But I don't want to leave you with the idea that there isn't some kind of base pairing here. I'll sort of draw a rudimentary structure where we can see that we begin to get a kind of three-dimensional shape out of RNA. So if this is an adenine, and this is, say, a cytosine, a guanine, and a cytosine. This adenine can base pair to can base pair to a uracil over here. This cytosine to a guanine, this guanine to a cytosine, and this cytosine to a guanine. So we'll get that base pairing. And then down here, where they don't base pair as well, there may be some other bases. But by having the molecule, even though it's single-stranded, by having the molecule fold back on itself and form this base pairing, we can start to get a rudimentary 3D shape of RNA. We'll see tomorrow this is particularly important in one type of RNA called transfer RNA, the way it's folded back on itself. But for now, just recognize that these RNA molecules can fold on themselves and they can produce a kind of 3D structure. Now we're limited in the 3D structure we can maintain or arrive at because we only have 4 bases to work with. But we do get some 3D structure, and this 3D structure then provides a kind of enzymatic potential. And what we find is that, in fact, some of these RNAs can act as enzymes. In biology, you'll find that one of the canonical places where we'll see this enzymatic activity is in the ribosome. And the ribosome, and a large ribosomal subunit that we'll talk about tomorrow and probably Monday, in those structures, we actually will use the RNA as an enzyme to form peptide bonds. But for now, just recognize that the molecule can fold back on itself, give itself a certain three-dimensional shape, and that can then arise with a certain enzymatic activity. So any questions up to there? Okay, let's go to the left-hand panel. And then let's over here begin talking about RNA transcription from DNA. I'm going to draw a DNA molecule up here, sort of a crude one like the one we had yesterday, but just to remind you what we were talking about yesterday. And we'll use the same kind of nomenclature where we have this. The phosphodiester linkages are shown by the blue line. And then there'll be these bases, and I'll just put a bunch of bases in here randomly. So this could be adenine, cytosine, guanine, adenine, cytosine, adenine, thymine, adenine, thymine, adenine, adenine, probably thymine, guanine, cytosine, adenine. Then there will be a complementary setup here, which will be thymine, guanine, cytosine, thymine, guanine, thymine, adenine, thymine, adenine, thymine, thymine, adenine, cytosine, guanine, and thymine. Now, one of the things I want to impress upon you, and first I'll give you the definition, and that is a gene is a sequence of bases in DNA that gives rise to an RNA transcript. But I'll remind you too that if this is DNA, and we looked at DNA in eukaryotic organism, this would be a structure that would go on for millions of base pairs. That will be an entire chromosome. And each chromosome may contain several hundred genes. And when you begin to look at this, it looks just like a scramble of individual letters. And the question that almost immediately arises is, how does the organism know where along this continuous string of bases, where can we say, how can we say, this is the beginning of the gene? This is the start point. Well, if a gene sequence is bases in the DNA, that means we're going to have several subsets of these bases within the chromosome that's going to produce an R transcript, which can, in some cases, produce a protein. How do we know where to start? Well, the answer is that a promoter is a sequence of bases in DNA where transcription factors bind. These transcription factors are proteins. So by definition, these are DNA binding proteins. Now, there are a number of different promoters, but the most common of the promoters is a 4-base set, said to be thymine, adenine, thymine, adenine. This is frequently referred to as the Tata box. And the Tata box is a consensus sequence of bases in the DNA molecule which provides a site to which transcription factors can bind. So what I've tried to do here is lay out a series of bases, but within that series of bases, I have TA, 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 and a couple more purines. You'll recognize this as the Tata box. This is the promoter. Now I'm going to label this because we need some orientation here. I'm going to say that this is the 3' end, which makes this the 5' end. And on this strand, because we're the anti-parallel organization, this has to be the 5' end. This has to be the 3' end. So what will happen is that there will be a transcription factor that will bind to this Tata box. This transcription factor is commonly something known as the Tata binding protein. If you get down into the weeds of this, there's some really interesting characteristics about these binding proteins. They have a particular structure called called a helix loop helix structure. And they have these 2 adjacent alpha helical regions of the protein. They have a kink in them, and they fit right down into the major and minor grooves. That's all detail. For our purposes, what the transcription factor is going to do is they have the specialized structure which allows them to bind to DNA, and once the transcription factor binds to the Tata box, to the promoter, then we recruit an assembly of proteins. That's simply to say that the Tata binding protein, which is often abbreviated as TBP, TBP, Tata binding protein. The Tata binding protein in itself is not enough to start the transcription. We need to bind the Tata binding protein to the DNA, and there's some clever structures to it. The Tata binding protein, in addition to that helix loop helix structure, has a series of amino acids which are positively charged, largely arginines and lysines, and they bind to the phosphate groups. And then there are some hydrophobic phenyl groups in there, probably from phenylalanine, and they'll insert down in between the bases and actually break the base pairing. So they open things up. But again, the Tata binding protein is not enough to start the whole process. It's essential, but it's not adequate. So that means we have to recruit another series of proteins, and I won't go into them. If you take a cell biology course, you'll find there are like 12 or 13 other proteins that have to bind to the site that makes a great big lump of proteins. This lump of proteins, which is in addition to the Tata binding protein, this transcription factor, the lump, let me be a little more accurate and say an assembly of proteins that are the transcription factor recruit RNA polymerase. So the sequence of events here is that we want to transcribe a certain region of the chromosome that will be the gene. We find the initiation point of that gene by tracking down the promoter, which I think it's about 60% of the time, is the Tata box. The Tata box is the site to which the Tata binding protein adheres. The Tata binding protein, once bound to the DNA, recruits this assembly of proteins, and that now recruits the RNA polymerase. So any questions to there? So now what I'm going to do is I'm going to go to the right-hand panel, and I'm probably not going to memorize this, but what I'm going to do is I'm going to lay out the strand, the template strand of the DNA. I won't put the complementary strand in. I'm only going to put in the strand we're going to read. And we're going to talk then about what that RNA polymerase can do. So we'll take this strand, and over here on the right-hand panel, I want to draw that DNA template. And I have to draw it in a particular fashion. I always have to draw it from 3' to 5', because what we're going to do here is we're going to read the DNA template strand, always, 3' to 5', and then we're going to generate this new strand of RNA complementary to that in the 5' to 3' direction. So this is the template strand of the DNA. This is the 3' end, and this is the 5' end. I'm going to put it in the Tata box. And then a whole series of bases down from here. Now, in fact, in reality, because the transcription factor, which is going to bind the Tata box, and let me put that in orange. So I'm just going to lump that assembly together here at the Tata box. And I'm going to label this as the transcription factor. Remember that it's recognized the Tata box by the Tata binding protein. We've recruited the proteins, and now we have this lump here. Now, in reality, this lump of proteins is so big that we don't begin transcribing the DNA strand until we're, about 25 or 30 base pairs downstream. That's the detail. What I want to emphasize here, and I'm going to tell you a bit of a fib and say that we're going to begin transcription right next door. That's not quite right, but for our purposes, it'll work fine. Because what I'm going to say is we're now going to take this molecule, this green molecule, I'm going to label this as the transcription factor. Sorry, I'm going to label that as the RNA polymerase. So the transcription factor recruits the RNA polymerase. The RNA polymerase now begins reading along the DNA strand from the 3' to the 5' direction. And what it does is it recognizes each of these bases. So I'm just going to put these bases in. I'm not sure what I have over there. I'm going to say a C, G, A, C, T, C, A, G, A, C, G, T. And what the RNA polymerase will do is it will find the complementary base. So the complementary base here for guanine is going to be a cytosine. So it base pairs those. It then finds the next base, running from the 3' to the 5' direction, finds this cytosine, puts in a guanine. Meant to put that in red. Puts in a guanine and forms the phosphodiester linkage. So this is the guanine. It enzymatically now forms that phosphodiester linkage using the energy and those extra 2 phosphates, now forming the phosphate bond between adjacent riboses, and I'll show that by the blue line. But each of the riboses, of course, is connected by the covalent linkage at the number 1 carbon to the base. It then goes down here to an adenine. Now the complement in RNA to adenine is going to be uracil. So we don't have any thymine in RNA, so the complement to adenine is going to be a uracil. The RNA polymerase finds a uracil, forms the phosphodiester bond, moves to the next one, cytosine will be a guanine, thymine will be an adenine, cytosine will be a guanine, and forms the appropriate phosphodiester bonds. And this continues. And I think you can see what we're doing. We're generating an RNA strand that is complementary to the DNA template strand. this will continue reading along the DNA, putting in one base at a time. But I want to point out that when this is generated, this is going to be the 5' end. And then down here someplace, it's going to be the 3' end. And all along that length, the RNA polymerase will have placed the right bases, complementary bases, to generate this strand. So adenine will be to a thymine, guanine to a cytosine, adenine to a thymine. Sorry, what am I talking about? This is RNA, so this has to be an adenine to uracil, adenine to uracil, cytosine to guanine, guanine to cytosine, thymine to adenine. So again, this is the DNA template strand. This is the newly synthesized RNA strand. That is complementary to the DNA strand. So what we've done here now is we've used the DNA strand, reading at 3' to 5'. We have found the Tata box, the promoter, in the DNA, bound the Tata binding protein to it, recruited the group of transcription factors, recruited from that the RNA polymerase, and then we began to read from 3' to 5' direction to generate a new RNA strand. So any questions to that? Okay, in the last few minutes, let me go back to the right, let me go to the middle panel, I have more room. And what I want to describe here are the types of RNA we're going to be dealing with. So the rules for making these RNAs are just as I've described. We need a region in the DNA, which is a promoter. We need to find that region in the DNA that's the promoter and bind a transcription factor to it, and then begin the process of RNA synthesis or transcription. But when we drill down a little bit farther, we find that not all RNAs are alike. And we find that there are basically 4 types of RNA. The first type, the one we're going to spend most of our time on, is called messenger RNA. And messenger RNA is abbreviated mRNA. And messenger RNA contains in its sequence contains within its sequence of bases the code for the sequence of amino acids. In protein. It gets its name messenger RNA because it's carrying the message of what the amino acid sequence is going to be. The second type of RNA that we'll talk about is called transfer RNA. We abbreviate this as tRNA. tRNA is effectively a shuttlecraft. I'll say it's a shuttlecraft that delivers amino acids to the ribosome. The third kind of RNA is called ribosomal RNA. And this type of RNA is the major constituent of ribosomes. It turns out that these ribosomes are not entirely made of RNA. They contain about 70 different proteins, and without those proteins they can't function. But the primary component, the major component of ribosomes is ribosomal RNA. We're going to see that these three are going to work in concert for protein synthesis. But there's a fourth kind of RNA that I never used to talk about, but it's with each passing day, this kind of RNA becomes more important. It's called regulatory. Some of these regulatory RNAs are involved in regulation of gene transcription. They can turn on and turn off genes. Some of them are involved out in the cytoplasm that help guide proteins to the rough endoplasmic reticulum. We'll talk in some detail about that. But there are a whole host of these proteins, and we're discovering more of them all the time, that are functioning as regulatory RNAs that are regulating the DNA and regulating other forms of RNA. We're going to leave those behind because mostly I want to talk about these three major types, messenger, transfer, and ribosomal, and how they work in concert to make proteins in the process of translation. And that's what we'll start with on tomorrow. So any questions? Okay, let's sign off then. We'll come back to translation tomorrow.